Okay, so we're live on YouTube. Hello, everybody. My name is Carsten Schradin. It's a pleasure for me to host today's Fine. And before I start with today's Fine, Eduardo will give you an announcement about what's going to happen in 2 weeks, because there's something special, unexpected that's not in the program yet. And he will give you a first idea now, and we will come back to it also next week. Eduardo?
Thank you, Carsten. Can you see my screen?
It is coming. Now we see it.
Okay, so very informal because we're just a community. I want to share with you because it speaks of the spirit of Fine. In the last 10 days or so, I've been in communication with some of our junior colleagues listed there. You know many of them because they're regular attendees to the meetings and they help us run in Fine. And we had a couple of meetings and I invited them to suggest ideas for a panel on May 23rd. And they came up with this wonderful idea. I'm sharing with you the text. I'll give you 30 seconds to read over it. This is what the panel is going to be about. So this is part of the letter that they wrote to a number of editors that I approached, and we're still working on this, but you can see now that we have already 4 editors in some of the leading behavioral journals who have agreed to join the panel. And the plan is that these younger colleagues of ours will be talking to me, and in collaboration with, they will come up with prompts and questions that they will pose to the panelists. So this is May 23rd. Please drop me a line if you want to be part of, if you're a student or a postdoc, you want to be part of the process of coming up with ideas for discussing during the panel. And we look forward to seeing you all 2 weeks from now. That's all. Thanks.
Okay, thank you, Eduardo. So just for everybody to understand, Heiko Rödl, who was supposed to present in 2 weeks, he had to cancel because of time constraints. And this is why we have instead in these 2 weeks, this discussion with editors about how to deal with statistics. So it's a pleasure for me. My name is Carsten Schradin, to be both your host and your presenter today. I'm a researcher at the CNRS in Strasbourg, France, working on social evolution in mammals. And today's speaker unfortunately also canceled, which is why I jumped in. But before I come to my talk, I would like, do you see the correct screen, Eduardo? The one of the tadpoles?
Yes, you're okay.
I first would like to come back to last week's talk by Chloe Folliot about exciting work on tadpoles in poison frogs, which you can see still on YouTube if you're interested. Very enthusiastic talk. And next week, I'm very much looking forward to a talk by Lee Corinne on Mars and Venus, sexual conflict over testosterone in wildlife. Lee is one of these colleagues that I know for a long time, only from her scientific papers, and she's doing exciting work on dassies, on rock hyraxes in Israel. So I'm also very much looking forward to finally meet her in person and hear about her work. And now, me, myself, no, that is wrong. What did I do? So.
We're seeing your desktop.
Yes.
Okay, there you go.
Oh, you should see it. Okay, so today's speaker unfortunately could not do it, and we said at some stage we should always be prepared to Fine present or Fine host if something like this happens, that we have a talk ready, which of course nobody of us ever has. But as it became clear that she cannot present, I had submitted an abstract to another symposium end of this year about the topic on harshness, and so I decided to jump in and to present to you today about harshness. So this is a talk I've never given before. Also, the symposium will be 10 minutes. Here I put it up to 40 minutes. My original talk is going to focus on ecophysiology, and I had to put sociality in. So my talk will not be...
Carsten, so that you know, we're still only seeing your desktop. We're not seeing, we saw for a second your first slide, but then we're not seeing it again. At least I am not.
Okay. You see it now?
No.
No?
Now I do. Now you're set. Yes. Okay. No, now I lost it.
Yes. I think it takes some time. That's why. So I think it should be coming up now. So this is only the Yes.
Now you're set. Now it's looking good.
Now that's the title, yeah. So I'm talking about harshness. I had to put sociality into this because we're fine here. The talk will not be so smooth. I never gave it before, and it was also not the aim. And it also made, the good thing for me is it also made myself think more about the concept of harshness and sociality, because my original work on this was more on the ecophysiological side. So this is based, and I also bring this for the younger colleagues to get some idea. This is based on a review me and colleagues wrote last year and submitted to biological reviews. And why is this pain this? And this actually was a review where we focused on the concept of harshness and 20 years of long-term studies on striped mice. But the editors of biological reviews didn't like it because they didn't like that we only focus on our long-term study. They didn't like the theoretical approach we were taking. For the theoretical approach, the solution was easy. We submitted it to another journal that is also not bad, which then published it beginning of this year, where we talked about harshness and why we think it's important to differentiate between harshness and stress. And the part on the striped mice is still currently under review, but this paper here is available. So we're here in fine. We're interested in social behavior, social systems, myself focusing on mammals. You see only mammals here on this slide. We have solitary living mammals. We have mammals living in groups, fission fusion societies, group living mammals without pair bonding and parental care, with pair bonding and paternal care. Here it's only maternal care, cooperative breeding, and so on. And we want to understand this biodiversity of social systems, like with the socio-ecological model, where we look at competition, at predation, at resource availability. And more recently, there has been also focus under which environmental conditions do specific forms of sociality like group living, communal breeding, cooperative breeding evolve. And in recent comparative studies, the concept of harshness and unpredictable environments became important in understanding this. And I want to give you a little bit a review of some of the most important studies here. And but before I come to the sociality, I want to mention this paper by Botero et al. In molecular ecology. That was one of the first ones bringing up the concept of harshness in ecological research, but they didn't look at sociality, but on species or subspecies richness. They had a principal component of climate variables for harshness that explain subspecies richness in both mammals and birds. And I'm also mentioning this paper here because many people later that worked on social systems used their databases, especially for environmental factors, to include within their own studies. Then from my point of view, maybe the most interesting or best studies have been done by Dustin Rubenstein's group. The first paper by Yates and Rubenstein in 2011, where they looked at cooperative breeding in birds. And they related it first to climate variables, that's temperature and rainfall, precipitation, and the variation of this. First in A and C among years, and here within years. So here we have seasonal variation. We see seasonal variation in temperature in Europe is given because we have cold winters and warm summers, while variation between years from one year to the other is relatively low because it's relatively stable and predictable, while in other areas it is less well predictable. And for rainfall especially, we see here in these desert areas, we have some years with a lot of rain and other years with little rain. So there's quite some variation, making it unpredictable for the organisms living there. Then they put this in relation to cooperative breeding in birds at different measures. Here is the total richness of cooperative breeding. Here it is proportional, and here only for passerines, and here for all non-passerines to show for robustness of the analysis. And we're putting together this occurrence of cooperative breeding and this variation in climatic factors within and between years. And what they found, we have here the graphs. So this is also, they did it for the different continents. Black is Africa, red Australia, then there's South America, and so on. And they, I'm not going into the details of the statistics, but they had something like a principal components and models of these climate variations. that put these factors together, and they found an effect of unpredictability, especially of rainfall, on the probability to find cooperatively breeding bird species. Especially what they found is that variables capturing environmental variability were stronger predictors than variables capturing environmental mean values. So the variability was more important, and the variation in precipitation, especially in rainfall, was also more important than variation in temperature. Among-year variation was relatively stronger than within-year variation. So if it's the seasonal variation, it's not so important than if there's a dry year, than a moist year, and this is not predictable. And biotic factors that were also in the analysis, like body mass, diet, bread, were less important than these climatic factors. So to summarize, they found it's not important whether the climate is moderate, subtropical, or tropical, but rather whether it's variable. It's not important whether it rains a lot or little, but whether the variation in rainfall is high. And differences between seasons were less important than differences between years. So this unpredictability, especially in rainfall, seems to be a driver of the occurrence of cooperative breeding. Then very interestingly and very nice was a follow-up study from the same lab of Dustin Rubenstein by Cornwallis et al. 2017. Now here, for the first time, they also mentioned the term harshness, harsh environments, and where they said that harsh environments are more likely to lead to cooperatively breeding species, both in invertebrates, mammals, snapping shrimps, and birds. And their study again was focusing on birds. We have here the phylogeny. In red are shown bird species with cooperative breeding, and in green is the environmental principal component 1, which they took into the analysis. Quite a complex statistical approach. I don't want to, and I cannot even present here in detail, but the main result was that there was a relationship between this principal component and the probability that a non-cooperative ancestor developed into a cooperative new species versus there was no such evolutionary transition. And this is summarized in this paper, and that's a very interesting point, was that they found that cooperative breeding from the analysis most likely evolved in benign environments, not in harsh environments, but that the probability of these species then to colonize harsh environments was very high. So in other words, we don't find a lot of cooperatively breeding bird species in harsh environments because they evolved there, but because they were better to colonize these environments from the ancestor that had evolved cooperative breeding in benign environments. So this was one of the first detailed studies on the effects of harshness or climate variation on sociality in birds. Then similar studies have been done on mammals. Here a paper by Lucas and Clattenbrock, where they compared between cooperatively breeding mammal species, so that's a breeding pair with non-breeding helpers at the nest, and monogamous species, so that's only the breeding pair and the offspring don't remain at home as helpers. And what they found is that they said, here we show that the distribution of alloparental care by non-breeding subordinates is associated with habitats where annual rainfall is low. So in dry environments, arid environments, it's more likely to find helpers at the nest. They didn't talk about harshness at all, but it's a similar study like with the birds, showing that climates and variation in climate and especially rainfall might be important for the occurrence of cooperative breeding. We did a similar study on marsupials that was published last year by my then master student Xingyu Qi, who is in the meantime a PhD student. She looked at marsupials and marsupial social organization, where among other things we found that they are less solitary as generally assumed. And I just want to pinpoint that we also found here some effects of climate and climate variation on sociality, again with principal components, looking at temperature and rainfall and variation between years and within years. And what we found is that group living species were less likely to be found in climates representing tropical rainforests, or in other words, solitary species are more likely to occur in the stable environment of the rainforest. And we were the first ones that took variation in social organization also into account. It's so-called IFSO, intraspecific variation in social organization. We found that species with a variable social organization were associated with low and unpredictable precipitation representing deserts. So when it rains little and it has a huge variation from year to year, it was more likely that the species showed variation. I also want to show you here that if you look at this, even though there's good statistical evidence for this relationship, when you look at it, at the data itself, this evidence is not very strong. So we cannot really predict by just looking at one species whether this here will be pair living or variable and so on, but at least it goes into the same direction. But there is variation that is absolutely not explained, also meaning we might have to come in future to a better and more precise definition of different factors than we did there at this stage. Last but not least, I want to mention the find that was given by Jordan Martin, I think 2 years ago, a PhD student at the University of Zurich in Switzerland, who showed in another paper, I won't show the results here in detail, that even in humans, harsh environments are related to the occurrence of alloparental care. So summarizing these studies, there's good evidence that harsh environments, especially with low rainfall and variable climate, favor the evolution of complex animal societies, especially cooperative breeding and helpers. So here, harshness might be important. What is also apart from social evolution, discussed in the literature is that increasing harshness due to climate change, such as extended periods of heat and drought, has been identified as a major threat to biodiversity and ecosystem functioning. Now, one problem here is, and you might have realized this by my problems formulating all these results, is that harshness is always used as a term which everybody knows, and it's not something good, we agree, but it has never been defined that very well. So reviewing this literature, there's no clear definition of harshness. People just use it and everybody understands it. This includes also papers by my own research group in the past, where we just used the term harshness as an everyday term. And we think then when we were thinking about this more clearly, that it might be important to clearly define this term, because to predict long-term consequences of harshness, we must identify which mechanisms evolve to cope with it, what our limits are, and when they might fall. So we all know there's global change. We expect environments become more harsh. It will be a problem. But to understand this, we first, from my point of view, have to come to a clear definition of harshness. Here you see a table with definitions or ways harshness was measured in past papers, like here chronologically ordered from Webb 1980, who was studying marmots with heat stress plus length of growing season. So it has something to do with food, but it's difficult to put this concept generally for all animal species. Then Botero, like the paper I showed at the beginning, they took a principal component analysis and put different variables like also we did with the marsupials into this principal component, rainfall, temperature variation within and between years, like was also done by the research group of Dustin Rubenstein. Now, this is how this theoretical or this opinion piece came into existence, where we argue that we must differentiate between stressful and harsh environments to understand animals' resilience to global change. And our proposition is that harshness is not stress. Stressful environments activate the physiological stress response to increase energy availability, while harsh environments inhibit the physiological stress response to save energy. In other words, harshness response and stress response are mutually exclusive. Now, I want to explain this to you in more detail. And here the first big question is that has followed me since I started to study biology. What is stress? Even there's no agreement. Most people think stress is something bad. It's threatening homeostasis, survival, reproduction of the individual. But how to define it? And for me, who works at an ecophysiological laboratory, the best and only suitable definition of stress is via the physiological stress response. For me, stress is when stress hormone levels get increased by something that's happening here, which is typically called a stressor. Something happens, maybe there's a predation event. These are data from studies on laboratory rats. Something bad happens to the rat, and then adrenaline levels increase immediately within seconds and then slowly decrease again. and levels of the HPA axis, so the hypothalamus pituitary axis, adrenal axis, the glucocorticoids, they increase after 3 to 5 minutes, then increase over a longer period of something like 15 minutes, and then decrease again. So for me, stress is when the physiological stress response is activated. And in contrast to this, working in a semi-desert in South Africa, harshness is when food availability in the environment decreases due to seasonal drought. So that's not something that happens in a moment, but it happens continuously, and not much is changing at the beginning with the glucocorticoids, but then over time they slowly decrease more and more and more. Why? Because glucocorticoids are not only stress hormones, they're important basic hormones that regulate how much energy is available for the body. For example, for diurnal species like us, in the morning, about half an hour before we get up, we have an automatic increase of glucocorticoids in our blood system that gives us energy that we can get up in a normal way. If you wake up 2 hours earlier because you have to go to an important meeting or whatsoever, it's much more difficult for you to get active during the day. And the hormones of the sympathetic system, we expect not to be affected at all. This is very important to show that this response is totally different. And this is also important to understand that harshness is not long-term stress or chronic stress like many people, including myself, automatically normally believe. They say, okay, harshness, it's a bad environment so for a long time, so it's long-term stress. But what we would find in long-term stress or chronic stress, these are studies. We took here the example of Tobias by what's his name no, I have his face in front of me. These studies that were done in Germany, where Tobias, a pair-living species, Dietrich von Holst, of course, did these studies, and they're pair-living species. And when you put into a pair a third individual, typically a male, an intruder, then this is stressful both for the intruder and for the pair male. And if this intruder is not removed, this stressor does not disappear. And what you find is that the stress hormone levels of the sympathetic nervous system, so the adrenaline increases a lot and they stay at a much higher level than before. And the glucocorticoids of the HPA axis, they remain very high until the intruder is either removed or what also happened, died. If the stress of the intruder is not, and this is then long-term stress, so the levels don't go down after minutes, but after days. And if this doesn't disappear, then it comes chronic stress with permanently elevated stress hormone levels, which of course is not healthy and also not adaptive. So that's another important point here to make, is that the time axis differs between these three patterns. When we have a normal adaptive stress response, it happens within minutes. There's a predator coming, you get a lot of adrenaline, you run away, your glucocorticoids increase, but then after half an hour, it's back to basal. But if the stressor does not disappear because you have a very, very annoying colleague or something like this that you see every day, then these stress hormone levels will be increased permanently. While in harsh environments, these hormone levels are, here we talking about something that happens over weeks or months, and here the hormone levels do not increase, but the animal has to find a way to endure this harshness, hoping that environmental conditions automatically become better at some time. In other words, the stress response happens so the animal can do something to escape the stressor, while the harshness response is something that must happen to enable endurance. So to summarize, this stress is the detrimental environmental event activating the physiological stress response. The physiological stress response is the activation of the sympathoadrenal medullary axis and the HPA axis, which activates energy to quickly overcome the stress. Subsequently, the baseline levels of stress hormones are restored via negative feedback. And stressful environments then are environments characterized by events triggering the physiological stress response, leading to increased secretion of stress hormones and causing energy mobilization, and this energy is used to overcome the stressor. In contrast, harshness we see as long-lasting environmental conditions which decrease energy availability, and harsh environments are characterized by persistent, long-lasting factors that threaten homeostasis and survival if the animal remains at a high energy expenditure. So the harshness response are behavioral and physiological responses to endure harshness. The harshness response of non-migrating animals, migrating animals like birds or some bat species, if there's no food, they might increase energy and fly away to get to better grounds. But non-migrating animals like small mammals, there is a decrease in energy expenditure. For example, we have a reduction in metabolic rate and activity, including downregulation of the physiological stress response. So the harshness response and the stress response are mutually exclusive. They are not the same, and as such, harshness is not long-term stress, which is the activation of the physiological stress response for extended periods. In mammals, this is about a couple of weeks. And chronic stress is the long-term activation of the stress response for more than 4 weeks, leading to high energy expenditure that depletes energy in its energy reserves without overcoming the stressor, which of course is not adaptive. And chronic stress, typically in the literature, is mainly described either in humans or in captive animals. It's not to be expected to occur under natural conditions. To make this a bit more clearer, I have here an example how it can look in my study area, the Succulent Karoo semi-desert, where we study the striped mice. A stressful event could also be the weather. In winter, when the rainy season, there are cold fronts coming in from the South Pole, from Antarctica, with a sharp decrease in ambient temperatures. We see here before it gets up 30, 20 degrees. Then the cold front comes in with cold rain, frost. It gets very cold, and these animals have a thermoneutral zone around here. When it goes down to 5 degrees or even frost at night, they have to spend a lot of energy to keep warm, while the cold rain does not allow them to go out for foraging. It's expected that to overcome the stress for the event, they have to increase the energy reserves for thermoregulation. In contrast, long-lasting climatic events cannot be overcome by a short-term increase in energy. We look here over months and years, and we have in green the food availability on the field side on this axis, and here in bars the rainfall. We see there's breeding seasons with high food availability, and then there's dry seasons with little rain and low food availability. These dry seasons differ in harshness. Here dark brown, there's very little food, and light brown, that's a year with relatively high food availability, and they must find different ways to cope with this. And this is the second part of my talk, talking about mechanisms of the harshness response in striped mice. So I'm doing field studies since 2001 at the Guchab Nature Reserve in South Africa. This is 600 kilometers north of Cape Town. It's a semi-desert environment with rain mainly in winter. It's an open habitat where you see it's characterized by these succulent shrubs. It's a photo from summer, and it's, like I said, a long-term study on sociality, and if I look at myself, maybe also on aging. So we have data in the meantime for more than 20 years. And I have been studying the striped mouse, a small diurnal mammal occurring in this open habitat, easily habituated to the presence of observers, which means we can observe them directly in the field because they're active during the day. And like you saw in the photo before, it's sandy patches, which normally is only possible in larger mammals. It's also flexible in its social organization. They can live solitarily or in communally breeding groups with 1 breeding male and 2 to 4 breeding females. A reason why I study this animal are the large sample sizes that are possible on a field site of 10 to 20 hectares. We always had around 12 study groups with 60 to 250 marked individuals, which we followed during their entire life history, and from which we could take repeatedly blood samples for ecophysiological research, hundreds of blood samples every year from 60 to 100 individuals during different stages and during different seasons. It's also nice because one can study species because studies in captivity are possible and relatively cheap compared if you study primates or larger mammals. And interestingly, from an evolutionary point of view, it's an annual species. They live for about 1 year. The breeding season is in the austral spring, starting in August, September. Then they're breeding until November, December. Then comes the hot, dry summer from January till April, May, where there's no breeding, followed by the cold, wet winter, where there's also no breeding, probably because it's too cold. And individuals born during this breeding season, when they reach the onset of the dry season, have a chance of maybe 30%, in bad years only 3%, to survive till the next breeding season, but nearly nobody survives for a second year. Out of 10,000 marked individuals, we had less than 60 that survived for a second year. So it's basically 1 year, 1 generation. Like I said, it is a seasonal environment, and spring is moist and cold. Temperatures at night can fall below 0. And for a small mammal that has a thermoneutral zone at 30 degrees, this is very cold. They have to spend quite some energy to keep warm during this period. And but it's then very green. You see everything that's green here, or orange and yellow. These are wildflowers. Everything you see here is mouse food. Everything. Then in the dry season, all of this disappears and there's very little food left. You see here some succulent shrubs at a dry riverbed that is here at the back. And these succulent shrubs offer some emergency food, not of good quality, but at least that food source is abundant, but it's probably not very good because of a lot of plant toxins in it. It is getting warmer, but even in summer, temperatures can go down to 5 degrees at night, 10 degrees, and this is still very cold for such a small mammal, because they're not nesting inside burrows below ground where it's warmer, but they nest in grass nests on the base of thorny shrubs, so they're exposed to the ambient temperature, and they still have to spend quite some energy to keep warm. Here we see 15 years of data. Again, green is food availability measured on 8 monitoring plots monthly, and black bars is precipitation, rainfall. And we see again here in the breeding season, there's lots of food. Then in the dry seasons, in summer, food availability decreases dramatically, and there's huge variation from year to year, which means from generation to generation. The mice born here had a season that grew up first when there was lots of food, but then they had a terrible dry season, while their children and grandchildren had a not so harsh dry season, and then their grandchildren again had a quite harsh dry season with little food availability. This red line here shows indication from our physiological markers when these mice start to get into a phase of fasting. That means when blood glucose levels are decreasing, ketone levels from fat metabolism is increasing. So everything that's below this line indicates there's a period of food shortage which these mice have to endure to survive from this dry season to the next breeding season and to reproduce. Here it was not very difficult, here a little bit difficult, and you see here this generation, they had to endure these harsh conditions to reach the breeding season in the following spring. For example, here we have, again, in the green line, food availability. This is all for only a little bit more than 1 year. And the yellow bars are blood glucose levels, which we measured in these mice, and they correlate nicely with food availability. And also interesting to see is when we look at May 2010 and May 2011, there's variation from year to year with much less food. In 2011, and at the same time also their blood glucose levels were significantly lower. By the way, as a reminder here, April 2011, that was the year when there was this one volcano that had erupted, making travel via air impossible. And this is why for this month we don't have data, because the manager who does the plant surveys was stranded in Zurich and could not return to South Africa. These are long-term data over many years, and we found a significant correlation between blood glucose levels and food availability. Again, there is huge variation, so it only explains relatively little, but there is this pattern that less energy in the environment is also related to less energy within the physiology of the animal. And even though this pattern is not very impressive, at least I can say that in the following years, this is data until 2014, in all the following years, we still found always this correlation between blood glucose levels and the food plants that we measured. And I can tell you already now, we find many relationships of physiological and energy measures with the food plant availability. These relationships are often not very strong and evident, but at least I can say that it's consistent over all the studies we have done. And if we would have a better measure of food availability in the environment, maybe we would get better relationships. I mean, these food plants are many different species, but at least we can measure the food availability to some extent. So another way that's very important for the striped mice to save energy is basking in the sun. Actually, sun basking for them is very important. We are in a valley, and the nests of these mice are basically switched on by sunshine. They are nesting on the base of shrubs above ground in grass nests. And when the sun comes over the valley, as soon as it shines on the nest, a few, not even minutes, less than 1 minute later, the mice are outside sitting in the sun and basking. And we were the opinion they don't do this just for fun, but it must have an adaptive value, and it might help them to passively heat up and to save energy. And this was studied by Mike Scantlebury, who is now at Queen's University in Belfast. At this stage, he was a postdoc in Pretoria. He came to the field site. We went to the natural basking spots. We put the mice in such chambers to measure their oxygen consumption. Here we have two examples of mice. We have first here, this line on top is the ambient temperature. It increases a bit when the sun comes up, of course. Then we have this small line is illumination, so lux, measured how much light there was. There was no sun, then the sun shines on the nest and it increases. And then after something like 10 minutes, we put a cover over the chamber to shadow it, and the light in the chamber decreased while the temperature was still, because it was ambient air, was still increasing a little bit. And here these dots is the oxygen consumption when they were not basking, because they were in the shade. Oxygen consumption was high. As soon as the sun came up, they could bask, oxygen consumption decreased. And as soon as we shaded them, oxygen consumption increased again, showing that they can significantly save energy by sun basking. In another study, we also showed that they are not sitting at random spots, but that they choose spots that are on average a little bit warmer than random spots. So it seems that they choose their basking spots depending on the microclimate. But most important, I think, for them, why is this video not working? I don't know. I even updated it today. What you should see here is a time-lapse recording where you see mice at night sleeping inside a natural nest. It looks very white because I provided them with some toilet paper outside the shrub so that they could mark the nest for me to find it. And they really form a ball of mice warming each other in the nest. And Mike was measuring from captive mice and their metabolic rate at night at different temperatures, from 5 degrees to the thermoneutral zone of 30 degrees on different group sizes. And what we find is that the single mouse that was sleeping alone always spent much more energy, except of thermoneutral, than the mice sleeping in huddling groups that could warm each other. And the temperatures of 5 degrees, 10 degrees, quite common even in summer. We see, for example, that the single mouse here had nearly double as high resting metabolic rate than the mice sleeping in huddling groups, and spending every night double as much energy is, of course, in an energy restricted environment, quite problematic. And I believe that the main reason why striped mice do form groups is actually because of these benefits of huddling to save energy at night in the food restricted dry season. Of course, they can also save energy by behavior. These are studies, and what comes now mainly done by Rebecca Rimbach, a postdoc who was with us, who is at the moment now on a contract position at the University of Münster in Germany. And here she, together with students, they had the undaunting task to follow striped mice during the moist and dry season for their entire activity period, from leaving the nest in the morning when they went out for foraging until they came back at night. So in the hot summer with 30, 40 degrees for something like 14 hours, following a mouse with a radio transmitter. And the students were always observing them for 3 to 4 hours, and then other student took over, quite demanding to get these data. And what she found is that in a dry season, this year's inactivity, they were much less active than in the moist season. And they also spend less time in the dry season foraging than in the moist season. That might be surprising because there's less food, but again, there is not little food in the dry season. There's everywhere these succulent shrubs, it's just crappy food. Yeah, they always find something to eat. It's just not good, so they don't need to invest a lot of time to find this food. But if they would run around in the sand, it's also not like they would find something very nice and juicy. It would be a waste of time and energy to spend more time foraging then. Looking at hormones, we found basically all hormones to decline in the dry season. Here, corticosterone, again, like I said, it's not only a stress hormone, but a metabolic hormone. It's known to be increased during the breeding season due to high energetic demands in both mammals and birds. And in here, 3 out of 4 different social categories, that is philopatric non-breeding males, breeding females, and philopatric females, it decreases significantly. During the dry season. Here they have high levels because of the cost of reproduction. Only the breeding males have low levels because high corticosterone levels would suppress high testosterone levels, which they need for spermatogenesis. So the breeding males have very high testosterone levels in the breeding season, which then decrease in the dry season. But even these 3 other categories show a decrease in testosterone from breeding season to dry season. Another hormone very important and most important for metabolism is triiodidtyronine, T3, which is known to higher levels increase metabolism. And here we also found seasonal differences with lower levels in the dry season. And again, here we find a correlation between triiodidtyronine and food plant measures. It's significant. It's again not that very impressive, but it's consistent with all the other measures that we have of energy consumption and metabolism and food availability. Another study also where Rebecca analyzed a hell lot of long-term data on body lengths and growth rates. What we found is that striped mice grow very slowly and very long. For males to be here's the onset of the dry season, and the males that survive until here, the bigger they are, the more competitive they are, and that can have a huge impact on their reproductive success. For females, larger females have large litters, so there is never a selection force to still grow, even though there's little food. But with that little food, they cannot grow fast, and so they grow for a very long period. Here, this graph here, this dashed line, is when they reach sexual maturity after 6 weeks, theoretically, and they still grow for a long period afterwards. And most mice, of course, die here during this dry season, but for the ones that manage to survive here and start reproduction, they don't survive too long either. And this here is when they, on average, about die. So that's the normal life expectancy of a successful mouse. And it shows to you that basically these individuals grow during their entire life. So this is quite unusual. This is like if I, as a human, would have kept growing until I'm 50 years old and only stop afterwards. So it's a long period. And we don't talk about growth in body mass, which maybe I increased until I got 50 last year. But we're talking about skeletal growth. So this here is body length, not body mass. We also looked at organ sizes. For some studies, we experimentally removed mice to reduce in population density. Then we used them to study organ sizes, and we found that energetically demanding organs like the livers and organs of the digestive tract, which are energetically expensive for digestion, they decrease in the dry season. And interestingly, we also found that the brains are, compared to body mass and the size of the other organs, significantly smaller in the dry season than in the moist season. And obviously the brain is an energetically very expensive organ. This is the so-called Dennell effect, described some decades ago in shrews here in the northern hemisphere, where shrews decrease their brain size in winter, probably to save energy. In the meantime, it has also been shown in some mustelids, in polecats, and this would be the first example of a rodent that there is a reduction in brain size in the food-restricted season, which in this case would be the summer dry season. Would also be the first study showing that this is not only occurring in winter in the northern hemisphere, but can also occur in summer in the dry season in the southern hemisphere. But what about energy expenditure itself? We measured resting metabolic rate. We have a permanent respirometry lab at the field station where we measured for many years 3 mice every day. And we found here, for example, in the year 2013, a reduction of resting metabolic rate in the dry season of 11%. Some people say 11% is not a lot, but I think if I experienced since many years a decrease of 11% in my research funding from my employer over years, and I can tell you it is quite a lot. And if somebody would give me a raise of 11%, I would not complain about that. And looking at all the long-term data, we again find this relationship between resting metabolic rate and food availability, but only for the dry season, which makes sense, not for the moist season. The moist season, as always, I showed you this, there's so much food, they just can eat as much as they want, and then they have the best resting metabolic rate, not too high, not too low, but it doesn't increase when there's more food. But in the dry season, it depends on the food availability, whether resting metabolic rate is lower or higher. And then last but not least, Rebecca also looked at daily energy expenditure itself using the doubly labeled water technique, with which one can measure how much kilojoules they spend per day, and found that there was a reduction in the dry season compared to the moist season of 40%. So that's quite a significant reduction in energy consumption. Then just quickly, to summarize this, so the Harshness response of striped mice, they save energy in the dry season by social thermoregulation in huddling groups, basking to passively warm up, reduced activity, reduced levels of metabolic hormones, reduced resting metabolic rate, reduced growth rate, leading to an overall reduction of energy consumption of 40% to enable them to endure this harsh dry season, to survive to the coming breeding season. And what about cognition? That was another project we had for many years, ecophysiology of cognition, mainly done by Audrey Mayer and later by Céline Rocher. And in theory, we expected there could be three outcomes. If there's less energy in the environment, then animals have to trade off what to spend the energy for. And cognition, the brain, of course, is energetically expansive, and it could lead to cognitive impairment, that cognition, like other functions, decrease because there's less energy available for the brain. Could also be that there's cognitive resilience, that cognition, being so important, is taking out of this trade-off and always gets the same amount of energy. Or even that there's cognitive enhancement if, by being more smart in this season, you can get more food. And which of these three mechanisms take place might be species specific, depend on the life history and on the ecology of the specific species. It might be different from one species to the other. The studies we did, we measured cognition in different ways. Here we have on this screen, we showed them a little photo of a predator. So that was then how much attention they pay to predator cues. Then we had here an elevated maze where we measured spatial memory. And here it's a pity that these videos don't work. I don't know what I did wrong. Let me see. I must. Now here it comes. There you see what these mice are doing. And here is problem solving. They have to open this lid to get to food. And these were studies done by these two postdocs. And they nearly always found an effect of season, but the effect was not consistent. Sometimes it was only for the males, sometimes only the females. Sometimes they were better, sometimes they were worse, sometimes there was no difference. And for me, it just means when you have a lot of data, you always find something, and it's no consistent pattern in contrast to what I showed you before. And for me, the best interpretation is that these striped mice have cognitive resilience, and their cognition is not impaired, but also not improved. So the harshness response of striped mice, resilience to environmental harshness through reduction of energy expenditure, which, by the way, also increases water retention, because a higher metabolism, the animal uses more water, but while the cognitive function is maintained via behavioral flexibility, passive thermoregulation, active thermoregulation, reduced activity, and physiological flexibility with all these patterns I have discussed before, like lowered blood glucose levels. We also showed ketone levels of lipid metabolism are increased, lower metabolic hormones, leading to reduced resting metabolic rate and then reduced overall energy expenditure. Putting this into a more general frame, it might also help for other species. It's important again to point out that this is mutually exclusive to the stress response. The stress response, the fight and flight syndrome, energy expenditure is increased by metabolic hormones, stress hormones being secreted, more energy to the body, and the animal can fight its opponent or run away, and with this overcome the stressor. But in the harsh environment, they have to endure it. They have to find a way to survive it until the environment itself changes. They cannot change the environment unless, again, it's a migrating species that flies away, or large mammals might also walk away. And they have to find ways to endure these conditions. Now comes the more problematic part. I would for this fine seminar like to come back about to sociality, what made me thinking the last 2 days. And these slides I changed about 20 times, and they're still not perfect and maybe not even good. But at least to show you also what we have here for the striped mice is we found in many studies and replicate in different ways that there's a relationship between population density and sociality in the breeding season. With high population density, most striped mice living in groups, low population density, all being solitary, and in between we find both tactics. And we interpret this and we show that there's reproductive competition, females killing each other, pups, males showing sexually suppressing other males. So when population density is low, females leave the group to start independent solitary breeding to avoid female infanticide. The males follow. But if population density is high, they don't have this option. But outside the breeding season here, and these dots, and by the way, these mice here are exactly the same mice like down there, but 2 months later after rainfall. Outside the breeding season, they always live in groups, and I think it's really thermoregulation by huddling, by social thermoregulation, because they can save a lot of energy by sleeping together and warming each other at night, which is very important in this food-restricted season. So the harshness or the low food availability also influences their social system. But it seems that females prefer to breed solitarily if it's possible, and we also could show that it's mainly the large females, but the environment for them is kind of unpredictable. We had a look on, this was, I think, data from 10 generations or more, whether a female that grows up in a communal group with 2, 3, 4 breeding females, or in a or is the offspring of a solitary breeding females, whether this condition predicts under which condition it will breed later itself. And also here we looked whether females that grew up in years with above average population density could expect they will also breed in a year with above average population density, meaning communal breeding is probably the best strategy, or whether they grew up in a year with low population density, whether this predicts they will breed in a year with low population density, so they should become solitary breeders. And the chance was 50-50, because the environmental conditions are so unpredictable. And here's not only food, but also what's difficult to measure, predation pressure. And in this case, it's simply, is there a wildcat on the field side or not, which might differ randomly from year to year. And a striped mouse cannot predict from the conditions under which it grows up, the conditions under which it will later breed. And this might also explain why they evolved social flexibility, that they can flexibly react to the current environmental conditions. So to summarize this, what it might mean for sociality, is it rather harshness or unpredictability that might form the evolution of more complex animal societies? So first, what I have to say is this unpredictability of harshness, this unpredictability is also a characterization of stress environments. We originally developed this framework in the framework of ecophysiology, stressful environment versus harsh environments, and for both environments, unpredictability is an additional problem and not a characteristic of one or the other. But I have the feeling from what I reviewed today that for social evolution, unpredictability might be more important than harshness per se, because that's what Dustin Rubenstein in his studies found with his, he took rainfall, for example, was a little or much rain, and how predictable was it? And it was more the predictability that influenced social evolution than whether it was a lot or little rain. So I have the feeling unpredictability might be here more important, but I still wonder whether in future analysis to differentiate between harshness and predictability of harshness, whether this might lead to a better insight. Still, I think it's important to study benefits of sociality under harsh environments. And for this, we need a proper definition of harsh environment that is different from stressful environments. And whether harshness or the unpredictability itself favor sociality, I believe, can then be best studied in comparative studies and not in single case studies like we've been doing on the striped mouse. But also importantly, when we look at harsh environments, not all the species living there are social or have complex societies. And in fact, many species there might be unsocial, even or solitary living. For example, I had a look at a list of mammals occurring in the Kalahari. That's an old book of Anderson with 90 mammal species, and about 30 of these are assumed to be solitary living and only 2 to have complex societies. That's the well-known meerkats and the Damaraland mole rats. And so what we see is that even in harsh environments, in unpredictable environments, species that do not have complex societies can be very successful. In other words, harsh environments do not lead to the evolution of complex societies because most species are not. But it's more likely to find maybe these cooperatively breeding species in these environments that might also should maybe influence in the future how we ask our questions. So if harshness makes it more likely for sociality evolve in harsh environments, this does not mean that sociality is the best solution, because many species there might still be solitary living. And that's something I think would be nice to understand better in the future. So, and then measures of harshness, if you do case studies on a single species, I think it would be nice to really measure food availability. We measured food plants on our field site. It always shows a nice relationship, not so strong as we might hope. So there might be maybe better measures, but then for comparative studies, it will be difficult to find food estimates for many different species. Here one might have to go back to climate, expecting when it's hot and dry, there's not much growing, there's little food, and when it's moist and not too cold and not too warm, there might be more food growing. So there are other open questions with this concept of harshness. It was only a very short opinion piece. Harshness, so that's like what I call in contrast to stressors, factors that induce harshness. We mainly talked about food restriction, but water restriction and heat might also be important factors that we did not look into detail at the moment. Also very important is to understand that even in harsh environments, stress will occur. A mouse living in an environment with little food will still have to have a stress response in the case it encounters a predator. And this might be an interesting future research field, how this might work under different environmental conditions. For example, we predicted in a study that there could be a decoupling of the sympathoadrenal medullary axis and the HPA axis. In other words, in harsh environments, we expect glucocorticoids to go down, but still the sympathetic nervous system to be able to respond very quickly, which is something that occurs in some pathologies in humans, and it's only looked at it from a pathological point of view, but there might be environmental conditions where it's adaptive for this to evolve. Normally, adrenaline levels or sympathicus activity and HPA axis activity are very well correlated, but under some pathological conditions in humans, this correlation disappears. Then what would be harshest for marine environments? Of course, I focus on what I've been studying in semi-deserts, but for marine environments and for other taxa, like insects, it might be something very, very different. But whatever it is, I think we should try to define it. With this, I come to my conclusions. First, I maintain that I think harshness is not stress. These are two mutually exclusive patterns, even though in harsh environments, stress will also occur. And to understand in how far environmental conditions favor the evolution of sociality, it might sense to differentiate between environmental stress, environmental harshness, and the variability and unpredictability of these two conditions. I think maybe in the future, if we differentiate between the two more clearly, we might come to a better understanding. And for me, still one open question remains. Why are so many animals living in harsh environments, not social, but solitary living? It's so nice to see these studies showing that harsh environments or unpredictable environments are more likely to have social, complex animal societies, but still many animal species there are successful after the same evolutionary time that are solitary living. So solitary living cannot be just a poor solution, but it must be a good solution for many species. With this, I'm finished, and I thank you very much for your attention. So being the host today myself of my own talk, I have to make my own discussion to remind you, if you have questions, then please type a question mark inside the chat, and then I will call you up and see if I can try to answer or respond to
Carsten, before, some people usually need to go, before we start talking about your presentation, do you want to mention again the presentation that's coming up next week, which is something that for those who may have joined us a bit later?
Yes, so, oh shit, no, I just closed it. So next week is, here I have it again, Lee Corinne, a colleague from Israel, is going to present on Mars and Venus, sexual conflict over testosterone in wildlife. And I assume it will be mainly about her work on dassies. So that's South African. English is rock hyrax in the semi-deserts of Israel. Okay, then Filippo, you have a question?
Yes. First of all, I really want to congratulate, to improvise, since you talk a very nice example of putting a talk together in a very short amount of time, as you told us, and I think it was very pleasant. I really enjoyed it. So thank you for that. The second is a kind of a broader question. I'm interested in resilience, and I just wonder if you made very clear the difference between stress and harshness. And harshness, in your case, mostly was low food availability, but at the end you mentioned that could be also low water availability or some other resource maybe important for the animals, because the thermoregulation was more something that the animals, yes, need to deal with the temperature, but was the difference between if the environment was harsh or was not harsh, how to do it. So I believe the heat was not the harsh per se. The cold weather, the cold temperature was not the harshness condition, was a modulator of the harshness. So, and I agree with you, it was very nice and elegant way to distinguish between stress and harshness with the physiological measures. But sometimes you may not be able to get all the nice physiological measures that you have. So my question is a bit broader. Say, if there is no physiological measure available to distinguish between stress and harshness in the way you presented, but the researcher is still interested in resilience response. Is it critical to differentiate between stress and harshness, or would you expect a similar response of resilience to either one of the two?
I'm not very sure to fully.
I'll tell you again. Yeah. But, so the simple question is, do you expect resilience responses different between stress and harshness, regardless of what it is, the stress and harshness, so on the behavioral point of view, not the physiological point of view?
Yes, I think it would be very different, because I think normally for stress you don't need resilience, but you need a way to overcome the stressor by either fighting it or running away from it, and then it should be solved relatively quickly. If now you have,
why is not resilience that? Sorry, that's my question.
because here the animal is doing something actively. It avoids it, it runs away, or it's
not resilience that.
Okay, this I don't know. Then you have to.
Okay. That's basically my question.
Then one has to define resilience as a new term. if one defines resilience like this, to avoid anything that is bad, whether it is stress or harshness. Yes,
as usually is how is defined, I believe.
Yeah, then it's for me maybe not a very useful term, because it's a difference if you are resilient by a stress response or a harshness response. The stress response is energetically costly, and that means the individual, for example, if you don't have physiological measures, you would expect it, if it lasts for a longer period, to lose body mass relatively fast. While the harshness response is passive, but it would also lose body mass, but much more slowly, to be able to endure the situation for a longer period. So if you cannot measure, if you cannot take physiological measures, then taking measures of body mass might help you to still approach this. But also typically for stress, because the energy expenditure is high, but under natural conditions for a very short period, you might not find a strong decrease in body mass unless it's chronic stress, but this you would not expect under natural conditions. This is normally only reported under artificial conditions in animals kept in zoos or in humans kept in offices.
Okay, thank you.
That's one thing I want to mention, as Filippo mentioned it, the cold weather per se, like he said, is not really a harshness, but it depends on how much energy is available in the environment. In the moist season, it's much colder than in summer, but because there's so much food, it's not a problem. Actually, mice, they don't bask much in the moist season, even though they could save energy with it, probably because it simply makes more sense to go out and eat, because you can get quicker energy by eating more food than by sitting in the sun. So we see less basking, even though it's cold in the moist season than in the dry season. And this can also explain why some females, especially the big ones, become solitary breeding in the cold moist season. But for them, the thermoregulation is not a problem, because they find enough energy in the food that they can easily cover these costs. Then we have a question by Kaya.
Hello, nice talk. Kaya Tambak, Hunter College. I'm a postdoc. I am wondering whether, instead of cognitive performance, the trade-off when the brain is shrinking in the dry season might be more giving up on learning ability. I'm just thinking about some studies on fluctuations in brain size in birds, where males will have a period of when their brains are larger, seasonally larger, they're able to learn new songs, and then they kind of solidify their repertoire, and that repertoire keeps, it is maintained even as their brains shrink for the next season. So it's the learning that changes there. I wonder if there might be a similar thing going on with the mice.
Yeah, it's an interesting question, and of course you point out that I have two opposing results in my presentation. On the one hand, I say the brain is probably decreasing, but cognition is not affected by this. That doesn't make too much sense. So if the brain would really decrease, something must happen, and maybe we just did not measure it yet. You're right with this, and maybe it is singing, because striped mice sing a lot, especially during the breeding season. They have very complex ultrasound songs. Another thing that changes seasonally could be spatial memory still, because in the dry season they're restricted to a relatively small home range, and they only disperse when the food availability increases again. When they're dispersing, maybe, but we didn't find differences in what the spatial memory that we were measuring. But you're right, if the brain is really decreasing, then there should be also some cognitive effect, which so far we could not catch.
Well, what I was kind of thinking was, it could be that the cognitive performance is maintained, but the ability to learn new, whatever it is, songs, tasks, areas, is impaired. So it's not necessarily a performance, that you can measure easily, but the expansion of a repertoire with potential for expansion.
I mean, in the Barnes maze, we measured spatial memory, so that is learning. And in the studies by Celine, I didn't bring them in detail. There was problem-solving tasks where they had to open lids, where they have to remember to go the left door or the right door. So they had to learn something. And we did not find consistent seasonal differences.
Okay,
Yeah.
thanks.
And Suleima?
Yes, hi. Suleima Tang Martinez. I'm retired from the University of Missouri in St. Louis. And Carsten, thank you again for preparing this talk on such short notice. I'm sort of in awe that you were able to pull this off. So thanks. It was a really interesting talk. You know, this is not a topic that I have thought a lot about. So some of what I'm going to say is sort of coming off the top of my head, and it may be essentially what I think you may have been trying to say. But as I think of this issue of harshness versus stress, I wonder if the way that it would make the most sense, and if you would agree, that we can think of harshness as a characteristic of the environment, so that there are various factors that contribute to harshness. So, for example, unpredictability of the environment, low temperatures, lack of food, and so on. But that stress is a characteristic of the animal. And that would be represented by the neuroendocrine response, by its behavior. And so harshness is the environment, the characteristics of the environment. Stress is a characteristic of the animal, and obviously they're linked, but they're quite separate things that we're looking at. So harsh environments can be stressful in the sense that they can cause a stress response in the animal, but a harsh environment, I don't think we can talk about a stress environment. I think we can talk about a stressful in terms of what it causes in the animal, i.e., it causes stress. So would this fit with what you were saying, or am I saying anything different than what you said?
No, it does not fit. I mean, we see
Okay.
the environment can either have something that is typically called stressors that induces increased energy expenditure via the physiological stress response. A stressor could be a predator, can also be not meeting the predator, but the predator is around, and the mouse smells the wildcat feces all the time, and we know their behavior and energy then changes. It could be a competitor, a neighboring big male. It could be a short threat by extreme weather events. And all of this leads to physiological stress response that leads to increased energy expenditure to overcome this stressor. A harsh environment, I created a term harsher compared to stressor, and nobody with native English speaker likes this term harsher, so I was not allowed to use it in the paper. I still use it here. Harsher is like a stressor that is something in the environment, like low food availability, that induces a physiological response, a behavioral response, like reduction in metabolic hormones, reduction in activity to save energy, not to overcome the harsher, but to endure it until it disappears. So
do you think that it would make sense to speak of the environment then as a harsh environment, perhaps if it's dealing with abiotic factors, and a stressful environment where you're dealing with biotic factors?
I do not think we can differentiate.
I mean, both can induce a stress response.
Yes, like you said, I mean, a cold front is abiotic and can induce a stress response, and low availability of food plants is biotic, can induce a harshness response. So I think the main question one has to ask is the easiest. Is it good for the animal? Is it adaptive to increase energy expenditure, or then it's stress, or is it adaptive to decrease energy expenditure? Then it's harshness.
Okay. Okay. So you're thinking of it more in terms of energy expenditure in both cases.
This is the stress versus the harshness response, and then the environment that triggers one of the two is either the harsh environment or the stressful environment.
Okay. Okay. I think maybe what I'm getting hung up on is that when you talk about a harsh environment, it's essentially describing the characteristics of the environment, some characteristic of the environment that can subsequently induce a stress response, some sort of a response or, energy related response. And in the second case, we're talking about a stressful environment.
Yeah,
So
but
you can
again, the
have,
harsh environment does not induce a stress response, but it prohibits a stress response. That's exactly the point.
I'm sorry, say that again.
A harsh environment does not elicit a stress response, but it prohibits a stress response. So the harshest response is opposite to a stress response.
Huh. Okay.
Yeah, so stress hormone levels, they decrease, they do not increase in harsh environments.
Okay.
To save energy.
Okay.
Because increasing energy would make it even worse for the animal. It spends more energy, there's little energy, it would make it worse and decrease the chance to endure the harshness to reach the new environmental conditions that are benign.
Okay, I think I see what you're saying. I think I need to think some more about this, but I think it's very interesting. And I do think that this differentiation between harshness and stress is really important.
But this is the main reason we published this paper, that people always talk about environmental stress, everything that is bad, but everything that's bad in the environment can either, the adaptive response can be a stress response, increase energy expenditure to escape this stressful environment, or it can be the opposite, that the animal has to save energy. That means to suppress the stress
got it.
To endure it.
Yeah, I see what you're saying. Okay, thanks.
Yeah. So I think it's especially the common use of the term environmental stress that's problematic, because what people call environmental stress can lead to two totally opposing responses. And Then Kaya has
that makes more sense now.
Yeah.
Thanks.
You're welcome. Then Kaya has a follow-up question.
In your graphs, it looked like the small intestines and the liver also reduced in size in the dry season, and which is interesting because they're also energetically expensive tissues. What I'm wondering is just mechanistically, even for the birds that I read up on, like what do they think? Like, is some of the tissue actually metabolized and used up for energy, or is there a decrease in blood flow? How are organs, expanded and contracted seasonally, mechanistically?
Yeah. First, like what you mentioned, decrease in the size of metabolic organs of the digestive tract, including the liver, and so this is very well documented in many species, reptiles, birds, mammals. So that is nothing surprising in our study. And it is in fact believed that this tissue is kind of metabolized to put back into the system, and that the main function is simply that a big liver uses a lot of energy, and they reduce the liver because a small liver spends less energy than a big one, and this can then also partly explain especially the reduction of resting metabolic rate between the seasons, because the energetic basal consumption of these intestines is a major contribution to the resting metabolic rate.
So then for the small intestines, you can imagine it getting a little bit tricky because well, first of all, I'm not sure exactly how you would metabolize, a part of the small intestine. I guess it becomes, the tissues become thinner or something, but you can't really kind of chop off a section of it and then add it on later. So I wonder, exactly what's given up in the small intestines, particularly because if you degrade it too much, then you're threatening your ability to absorb nutrients efficiently and so on.
Yes, that's right. I'm not a specialist in this, and I read quite a number of papers on this topic, but I didn't read anybody really measuring what the reduction of these in small intestine sizes, how this is taken up. So I do not know, and I do not know if anybody knows. Yeah, it's an interesting question, but it's people just argue the way I argue here, but probably nobody could really ever measure it directly. Because I mean it's a slow process. It doesn't happen from one week to the other. It happens over weeks and months, and then it's difficult to really catch this up, I suppose.
Yeah, maybe you could do it with some kind of scanning if you had some, if you could bring them into a lab or something. But yeah, tricky. And
with this, you could maybe show how it slowly gets smaller, but to measure the metabolic consequences of this and how it's taken up. Maybe one could do this in a petri dish if somehow one can have a small intestine and make it grow and measure. I have no idea, but yeah, I don't never read.
They do have, they do have like equations that you can use in terms of like what the absorption rate should be based on like the length of the intestine. But again, I don't think that's what's given up. I think it's probably tissue thickness or something. But there are studies on, nutrient absorption based on all kinds of factors. I wonder if we could understand and measure, what part is given up. I think that there would probably be some theoretical basis for understanding how that affects absorption.
That's possible. That's interesting. Okay. Any other questions by anyone?
Yeah, I have a question.
Yes, David.
I'm David Wood. I'm a fourth year PhD student at Yale working with Eduardo. I think this idea of harshness is super interesting, and I'm wondering what role like environmental temporality or predictability plays. If harshness is just going to be defined as some sort of like input from an environment that leads to like less energetic expenditure, I wonder if that, I don't know, I guess it's a pretty open-ended question, but how do you think we should think about harshness in terms of temporality and predictability?
Yeah, I think that we took the predictability, variability out of the definition simply because unpredictability is also important for stressors. Yeah, I mean, these are also unpredictable. That's not the difference between the two. But for social evolution, I think that in how far, and that's maybe in future analysis, if they can differentiate between harshness per se and how predictable it is. I think, and that's the studies from Dusty and Rubenstein suggesting that the predictability might be more important than the harshness. And that also makes statistically sense because you don't find cooperatively breeding species in the Sahara Desert that is very harsh, but it's very predictable that there's no rain at all. But you will not see meerkats running around there. So if you have the harshness per se, at some stage, the environment will get so harsh that I think only a solitary species or many solitary species can be successful. But in the ones that are harsh, but not too harsh, their sociality might be beneficial, and how beneficial it then is might actually depend on how predictable, for example, rainfall is. Like the Kalahari and the Succulent Karoo where I work are relatively predictable deserts compared to some other desert areas, and maybe we have some highly sociable species here, but not so many. And it might be interesting if one compares over a broader range of maybe harsh, semi-harsh, arid environments, whether then the predictability plays a factor in social evolution or not of this harshness. Yeah. And that's maybe also the reason why predictability in these studies is more important, because once it gets too harsh, you cannot run around in big cooperatively breeding groups anymore. There's just too little resources. And Pedro wrote something about, I'm reading this here. I want to express my gratitude, drawing space. Dustin Rubenstein gave a couple of years ago, and it was mentioned today. Okay, there's a question. Okay, no, it's just saying thank you. So thank you, Pedro, for writing this, but I don't see a question here.
I'll be happy, Pedro, if you want, si querés preguntar la pregunta en español, con gusto, rápidamente la traducimos. I'm saying I'll be happy to translate if there's something that you want to bring up. Si hay algo que, Pedro, vos querés comentar o preguntar, con gusto, metele que yo traduzco.
Bueno, muchas gracias, Eduardo. No, pues no, la verdad no quería preguntar algo, sino de verdad agradecer porque la investigación es muy relacionada con lo que estoy investigando y me está
¿Podés
dando
presentar
muchos...
así? ¿Me podés decir, podés presentarte en español y lo traduzco así saben de dónde venís?
Ok, yo soy Pedro Ravelo, estudiante de maestría en psicología del Laboratorio de Aprendizaje y Comportamiento Animal de la Universidad Nacional de Colombia.
So Pedro is saying that he's very interested, was very interested in the talk because it's very related to what he does. He's a student in the master's degree in the lab of, if I got it right, psychology and animal learning. ¿En qué universidad, Pedro?
Universidad Nacional de Colombia.
In the National University of Colombia. ¿En qué ciudad estás, Pedro?
En Bogotá.
He's in Bogotá. So he finds your research, Carsten, very related to what some of the things he's doing. Digo que, bueno, traduje lo que decías, Pedro.
¿Y
sí,
qué
sí,
estudias, Pedro? ¿Qué estudias?
Soy psicólogo, estudio maestría de investigación en psicología y pues mi investigación es alrededor de que diferentes formas de interacción social como comportamientos cooperativos, altruistas y competitivos podrían considerarse como rasgos funcionales en respuesta a variaciones en las condiciones ambientales. Algo así como si los...
humanos. Dame un segundito. So, Sulema asking what you're studying, and Pedro is saying that he's a psychologist by training, and he's studying how cooperation, altruism, reciprocity could be functional responses. I mean, the relationship between those patterns of behavior or functional values of behavior and their relationship to the environment. So in a way, I think that if I'm getting him right, his work is very related to your last part of the talk, Carsten, where you're connecting sociality to environmental components. Digo que,
en humanos, Pedro. En humanos.
No,
no.
Okay. Pues hasta el momento he desarrollado como la parte teórica y pues no he encontrado alguna especie para poner a prueba las hipótesis todavía.
Okay,
he has been developing his theoretical framework, but has not settled on a system yet.
Okay, thank you for the positive feedback. So if there's no more question, I'm going to stop the YouTube. On YouTube, there's also no question. I didn't say it, but there's anyway only 5 people watching.